This is Steve Plotkin, and my poster is on epitope prediction for oligomer selective antibodies in both tau and A-beta. Okay, well, several previous in vitro, in vivo, and neuroimaging studies point to AD pathology as an A-beta exacerbated tauopathy, wherein soluble misfolded species of both tau and A-beta can propagate through the brain in a prion-like fashion. So we would thus like a method for generating antibodies to tau and A-beta, where those antibodies are conformationally selective to the propagative misfolded oligomeric forms, and so they would also have low affinity to isolated monomers, or particularly for A-beta, low affinity to fibrils and plaque, to avoid target distraction and ARIA-related complications. Our hypothesis is that regions of protofibrils that become solvent exposed when a protofibril is subject to partial denaturing stresses, such as what's shown here in figure 1, they will also be solvent exposed on the surfaces of toxic oligomers. Our method to predict these regions of low thermodynamic stability is to challenge the protofibril structures with a collective coordinate global bias that's implemented in molecular dynamics simulations. Following that procedure for A-beta yields 6 epitopes that are denoted here in figure 1. Applying the algorithm to tau allows several epitopes to be identified. To raise antibodies, we scaffold the epitopes using cyclic peptides of an optimal length such that the cyclic peptide best represents the conformation presented in the oligomer model, and that's shown in figure 3. The antibodies thus raised are conformationally selective. They bind cyclic peptide over linear peptide. They bind oligomers over monomers. And likewise for tau, they bind tau oligomers preferentially over monomers. Immunohistochemical staining of AD brain sections shows no binding of an A-beta oligomer selective antibody PMN310 to plaque, in contrast to other antibodies in clinical trials. Plaque binding is diffuse brown here. The dots are nuclei stained with hematoxylin. PMN310 neutralizes the effects of A-beta oligomers, increasing the viability of cells in an in vitro MTT assay. In an in vivo novel object recognition assay in mice, PMN310 completely prevents the loss of short-term memory formation caused by toxic oligomers. PMN310 preferentially binds the low molecular weight fraction of AD brain extract that's enriched in toxic species, in contrast to aducanumab and bapineuzumab, and tau oligomer antibodies raised to our predicted epitopes block aggregation as seen in an in-cell FRET assay.